Well, welcome all. Hello, and welcome all to the NIIMBL program webinar series. Today, I have the pleasure of sharing and introducing the N-mAb case study. My name is Sandeep Kedia. I'm really your host for this N-mAb webinar. Before we get started, I would like to make a few announcements, and we'll get started soon. So just to let folks know, this webinar is being recorded. Participants will be muted throughout the presentation. Please use the chat feature to ask any questions or post a comment. We will do our best to address the questions. However, if we don't get to our questions, we'll do our best to follow up after the webinar is complete. This webinar is open to both NIIMBL members and non-members. Please do not post any NIIMBL confidential proprietary information in the chat. Lastly, I'd like to remind folks there will be a survey link that will be sent out by email following the webinar. If you can complete the survey, you will have immediate access to the webinar recording. The webinar will also be posted to the NIIMBL N-mAb page later this month. Now, let me introduce to you today's speaker, Gene Schaefer. Gene serves as a senior fellow at NIIMBL, where he led the N-mAb case study and leads our regulatory committee, among other things. Gene joined NIIMBL in 2021 after over 35 years of experience in biopharma industry. Most recently, Gene was a senior director in API large molecule pharmaceutical development and manufacturing sciences at J&J. He also led the protein therapeutics development group at BMS and spent some time at Schering-Plough and Genzyme. Today, Gene is going to share with you the highlights of N-mAb, a case study to support development and adoption of control strategy for integrated continuous bioprocess for monoclonal antibodies. Over to you, Gene.
Very good. Thank you, Sandeep. And good morning, good afternoon, all. As Sandeep said, I'd like to spend some time today giving you an introduction to the N-mAb case study. So the N-mAb case study for integrated continuous bioprocessing is really just part of an overall NIIMBL strategy to develop case studies to support advanced processes. The N-mAb case study can be found using that web link or this QR code where you can download the full case study. NIIMBL also supported the development of A-gene and A-cell, which are case studies for gene therapy and cell therapy, respectively. And those were produced in collaboration with the Alliance for Regenerative Medicine, and NIIMBL provided a good deal of the funding for that. We have included links for those two case studies as well on that N-mAb page. Excuse me. That's part of the overall NIIMBL website. But this is one of the public-facing pages within that website. So, for those of you who don't know, N-mAb is a case study developed to really teach to integrated continuous drug substance process development. The second objective was really to help reinforce and standardize vocabulary, process definitions, and we provide a number of references in one place that would help anyone who's trying to start developing a integrated or continuous process in their organization. As part of this, we described two major process options with or without an intermediate batch hold tank so that we could discuss how you develop the overall control strategy, including adventitious agent control, and elements of managing that process in real time, including deviations. And I always like to put the acknowledgements up front so we don't forget folks. It was a large group of people who provided at least some input into the document. The leads are highlighted in orange, and many of the people that you see here were very actively involved in the chapter teams. Again, one of the real benefits of these case studies lies in the conversation that goes on in those sub-team meetings. And this is similar across the NIIMBL-led programs as well. I'd also like to acknowledge our tech writer and editor who helped us put everything together, Anna Hagen, and Jen Mantle did the overall production and design. So it is a very good looking document as well as one that has great content. So this slide is going to be very busy. I'm not going to go through this in detail. And in fact, a number of the slides today that I'll show you are really just introductions to invite you to go into the case study and explore further. So we started with an overall view of development of a biopharmaceutical manufacturing process and its transition to commercial manufacturing, various swim lanes around process design and validation, down through integrated control strategy. And then we mapped various chapters against those elements. Here's the overall table of contents. It is a fairly lengthy case study, about the same length as A gene and A cell, with quite a bit up front around the process design elements that go into the process definition. So we started with a comparison between batch and continuous processes, and not so much in economic or overall productivity terms, but really in how you manage that process. And one difference certainly is the number of intermediate pools present and how that impacts the overall dynamics. So if you look at the dynamics of a batch process, typical process, let's say it's maybe a 6 or 10 pack of bioreactors feeding a single downstream suite. At the beginning, say T equals 0 hours, you've harvested your first batch. 24 hours later, it's probably moved down to that pool past the capture column. Another 24 hours, at a reasonably quick cadence, you're already harvesting bioreactor number 2, and batch number 1 is somewhere in the polishing chromatography stage. Another 24 hours, you're getting close to the final viral filtration, getting ready to diafilter and buffer exchange that batch number 1. Batch number 2 is moved post-Pro A. And then again, another 24 hours, you have a third batch that's just been harvested, and batch number 1 has finally reached the drug substance stage. Now compare that with a continuous process. And here we're looking at a process with no intermediate holes, just a variety of surge tanks in between the individual unit operations. Okay, time equals 0, you're starting to harvest material. And again, note the difference. In that previous depiction of batch, that was a solid bar indicating a homogeneous concentration. Here, as you're moving material through continuously, you will see some variation in concentration of material, concentration of impurities. And as you move further downstream, that dispersion along the process path increases. And we've tried to show that with this sort of shaded bar here. And here, 6 hours compared with 24-hour in batch, you already have material moving past the capture step. Another 6 hours, it's moving downstream into polishing chromatography steps. 18 hours, you're getting ready to go through viral filtration. And 24 hours, the time in a batch process, you're only past the capture step at best. You're already collecting material for drug substance. Another 6 hours, you're stacking those individual aliquots that have moved through the process in that combined drug substance. So the dynamics are very different. The amount of material and its concentration will vary across the process, and the ability to control requires faster decision-making or controls. So coming back to a definition of batch, this is a slightly amended version of what's in the current case study on the website. There will be a version 2 coming out early next year where we've updated this definition, as you see here, based on some comments. But the definition of batch for this case study really goes back to ICH Q7 and the Q13 drafts. So it's defined by a specific quantity of material, whether input amount, output amount, or time of processing are used to define that amount of material. But as an operational definition for this case study, we've also added that you need to have a predefined amount of information and data set that goes with that material that enables disposition by the quality system, forward processing to drug product, and eventually release to patient. So again, it's important that the batch is defined by a quantity of material, that obviously can be a range, but also a pre-specified package of information. And that's very important for some of the points we're going to hit on later around control strategy and managing the process in real time. So we looked at two major options for discussion in the case study, whether to include or not a batch pool tank in the middle for collecting material and providing some homogenization and consistency for testing. And we discussed those two options and used the differences to highlight how you may make different decisions around how you would operate the process. Again, we tried to take a merged version of the type of continuous processes different organizations are running and present something that represents a certain amount of reality. And we also provided some definitions for the different types of surge tanks, really based on their volume and residence time. So a simple surge tank has a very short residence time. It's really more of a break tank between operations. A cycle surge tank has a longer residence time, and it accumulates one or more subcycles within a unit operation. And finally, there's a batch pool tank, if it's used, where you're accumulating material equivalent to the mass of a single drug substance lot. And that residence time could be 24 hours or more. And it would obviously require a larger vessel and probably some demonstration of stability over the length of hold in that tank. Now, along with those options, we did provide some detailed tables looking at how those options would play out for the different operations. And I've just shown the first 4 stages here and some of the differences that we've highlighted with pool tank or not, and additional options that were discussed. So, for example, in addition to continuous multi-column chromatography, we talk about operation in a dual column, alternating column load type of mode. Similarly, inline viral inactivation in a continuous reactor versus alternating batch tank pH incubation. Again, to try to capture the different options and variations people would want to use as they try to fit this process into a facility where the process is headed. And we talk about control strategy, how it's based on any prior knowledge, definition of the TPP and QTPP, that's the target product profile and the quality target product profile. This goes into risk assessment number 1, typically done prior to process characterization, and the outcomes from that are usually an initial control strategy for those first clinical batches. Most companies these days are operating on a very accelerated timeline. They're not waiting until process characterization is finished. They're moving forward into the initial clinical runs based on platform knowledge and some initial studies on the product. The second major adjustment to that risk assessment is typically done prior to process performance qualification. And the outcomes here are an updated control strategy that would be in place at least for the PPQ batches. In many cases, that is also in place for clinical material that's destined for pivotal trial use in the clinic. That way, there's a continuum between your pivotal material and the process validation, depending on how you may want to accelerate later stages of validation. And the third major risk assessment is typically done post PPQ, usually after you've answered questions from the initial set of filings to health authorities. And this is the beginning of your commercial control strategy. And it will also contain then elements of a CPV plan, the continued process validation or verification. And that package usually comes in 2 stages. The first set of batches, then finally after about 30 batches. And we try to emphasize that there's a definite learning cycle that goes on between what you develop during process characterization and validation, learnings from routine process control and monitoring, and non-routine assessments, including investigations, health authority questions from filings or inspections, and additional product studies that are done to support extended stability, whatever. So it's important to take this totality of understanding along with the process as it's moved into commercial sites. And then we talked about the control strategy elements, and we looked at an integrated control strategy for both process and analytical control. Usually at the beginning, there's very little information. A lot of that process capability is inferred from platform and process stress studies done up front. Process parameters are usually based on platform values, platform understanding. And many of the more advanced analytical testing, analytical stress studies, accelerated stability, not yet been done. So they're really here in outline form as placeholders for later updates in the control strategy. We didn't look at all potential quality attributes. We looked at a handful, various glycosylation attributes, deamidation, high molecular weight substances, and tried to build in what that process control and analytical control strategy would look like and how you would assess residual risk. It's interesting that, A-mAb has been very successful in that pretty much everyone in the chapter subteams had a great understanding of how you proceed with process characterization and validation. How you actually express a control strategy in a succinct written form that includes elements of both process and analytical controls, there still seems to be a certain amount of variation across organizations on this. So this could be an opportunity in the future for further harmonization. And then there are tables in the case study talking about the pre-process characterization risk analyses and how those would drive individual studies, their DOEs, one factor at a time. And again, with the understanding that a control strategy is a buffer against the effects of known variances. And these studies are where you explore the known variances. And we generated some data sets looking at, a couple of examples, say a dynamic perfusion, a steady state perfusion, how this would lead to differences in the downstream in terms of, say, titer coming into the capture column, how you would manage that. We also generated some prototype data sets looking at variation in quality attributes. Some attributes which remained relatively constant over time, in this case, the glycoform G2F, and in some cases, a glycoform, high mannose glycoform, which varied in one case. So for the G2F study over time, we included some analyses, which included typical sort of heat map of conditions that would lead to control of that attribute, as well as some Bayesian probability curves on what it would look like over time at different pHs and the likelihood of being within a specific range over time and how you could use that to drive control. We also looked at the case where you have a more variable quality attribute over time in the bioreactor and generated data, in this case, as a function of perfusion rate, so that lower perfusion rates gave higher levels of high mannose, lower viabilities, and then did some Bayesian modeling to look at probability predictions, probability envelopes really, at different perfusion rates for that high mannose attribute. And also included a very nice summary of different approaches to assessing time-varying attributes in the production phase. Again, these are really all too small to look at in depth here, so I do invite you to go into the case study and look at this. The analysis section is really very nice, including both traditional and Bayesian types of analyses. And we talked about process options a little bit more, considering this variability of one of the attributes. So since we were seeing increased variability of high mannose in one scenario, the dynamic perfusion, which is just as an example here, we talked about mitigating that variability through the use of a batch pool tank. That would be option 2 of the 2 continuous options. But we also introduced data related to impact of increased hold at that point in the process on deamidation rates, and made an argument that holding the process midstream would mitigate the variability for high mannose, but would increase the variability in the deamidation, and possibly to an unacceptable level. So then we presented a second option to mitigate that variability by choosing to run the perfusion in a steady state mode, which allows for a continuous downstream process with no long intermediate holds. And again, the data driving these decisions was manufactured for this case study. But it does represent the type of decisions that often need to be made and the types of data that need to be considered when making those decisions. And again, looking at that option that was chosen, option 1, with an integrated continuous process up until the cycled TFF step. This is what the process performance attributes and quality attributes look like over time based on our prototype data set for cell density, titer, high molecular weight species, and the high mannose attribute. And we then included additional discussion of the downstream considerations if you have a process flow that looks like this, including breakthrough curves if you're using a delta UV based load control, and different ways to approach the titration in the continuous viral inactivation step to make sure you're not titrating against a peak that comes through out of the previous chromatography step. Okay, we're about halfway through. Why don't we pause here for a few questions? Sandeep, do we have any questions?
Yes, Gene. We have one question. Can you comment on what are some consideration unique to integrated continuous bioprocess with respect to for process design and characterization and or validation?
Yes. So that's a very good question. So as far as process characterization, we did discuss for the most part how typical batch unit operations at the lab scale can be used to generate the data to support a fully continuous process at scale. So we weren't advocating that you necessarily need to run a lab scale process in a fully continuous mode. I mean, that would be nice, but you can generate the data you need from lab scale equipment that is already existing or, with some modifications in some cases. Validation also brings up some questions around how you really want to consider the validation set. Do you want to look at multiple lots in the downstream from a single bioreactor? Do you want to use multiple bioreactors, pool material into a single downstream? For example, if you're running at a site where you have multiple perfusion bioreactors operating in parallel, are you going to be pooling those? So we talked about some of those considerations. And then what we'll be talking about a little later is the consideration I mentioned earlier, the fact that there is a dynamic, that you need to worry about flow rates, you need to worry about the process dynamics, that you may only have a limited time to make decisions. How's that?
Thank you, Gene. There's another question. Was the model data informed by actual manufacturing experience? And how representative is the model data?
So the model data, as far as I know, does not represent a single manufacturing lot. It was informed by the experience of a number of individuals who are familiar with operations.
Thank you.
And I guess a question that, if people want to leave a comment when we send out the survey, we were considering including an appendix in the next version with that data. It would be good to know if that would be useful to people or not. Because we do know that A-mAb, going back, A-mAb was used extensively as a teaching tool in a number of universities and within organizations. So whatever we can do to make it more useful in a teaching mode would be very helpful.
Thank you. And I'll take one final question here. Does or could the control strategy incorporate product attribute measurements during the process and potentially reduce the amount of drug substance QC testing?
Excellent question. We'll come to that in a few slides, but the general feeling is yes. We didn't really talk about real-time release testing. There's a little bit of discussion of that in the future directions. That wasn't really the intent of this case study, but it could be, a supplementary discussion.
Okay.
And I know there are also questions that came up around model-based control, whether you can look at release of certain attributes based on parametric control of the parameters and attributes that control those attributes. We didn't really dig deeply into that topic within this case study. It was getting pretty long already, but that certainly could be a subject of follow-on work.
Okay. And Gene, if I can take one last question here, which is relevant. Can you discuss the use of plug flow reactor for virus inactivation and the difference between a PFR and other surge break tanks?
Well, in the plug flow reactor, and again, you're talking to someone who has much more experience on the upstream than the downstream side, but I'll do my best. In the plug flow reactor, you will have to have a much more precise control of residence time distribution through that reactor. Again, usually enabled by the use of mixing chambers up front, ensuring the flow rate is correct. You need to look at flow patterns across the front as it moves through that tube. That's the main consideration. The surge tanks may not have the same control over residence time because you may have variable inputs and output rates. They just need to make sure they stay within a minimum maximum operation rate based on agitation. There is some good discussion within the case study about the plug flow, as well as a number of references. And the last point, we did, as I pointed out earlier, look at the other option of just running that as, say, a dual tank alternating mode, where you're essentially running it as a cycled batch operation in the middle of the process. Some of those defaults, similar to cycling between two columns, some of those approaches are good gateways into a more continuous and integrated downstream if you're not ready to have everything fully continuous from day one. Okay.
Thank you, Jim.
So again, relevant to some of the questions, we did have a short discussion in that case study on operation at scale, including the potential for both offline measurements and inline measurements, and more discussion of process dynamics, the need to manage startup, shutdown, and response to any transient changes. Mass flow management related to process flow rates, surge tank sizing, potential for variance associated with certain operations, again, as came up in the previous question. As also good reference to the Kingman formula. Thank you, Jeff Baker, for bringing that up, which really comes out of queuing theory and talks about the impact of variance as a function of overall equipment utilization. And at some point, if you try to push utilization too high, small variations can make things very unstable. Excuse me. Trying to avoid the flu here. We also emphasized that the primary inline process sensors need to be robust, reliable, accurate over the expected runtime. And we did link to a very nice ASTM reference with very specific considerations around inline process sensors, and talked about that relation between sampling, testing, and decision-making, and what does the overall time lag in there, and how much time do you actually have to make decisions. And again, to go with those sample points for either inline or offline measurements, we put together a prototype sampling plan with some risk-based sampling frequencies and what you're really looking for. Again, probably too small for you to see, but I encourage you to look in the case study for a longer explanation of this. And as I mentioned before, that initial experience at scale or at a representative scale should lead you to an updated control strategy that represents a point at which you can go into the manufacture of material for pivotal clinical runs, clinical trials, or process performance qualification. And here you now have some understanding of the source of variation in the CQA, so the glycosylation CQAs. The source of that is the bioreactor. They're not stability indicating. There's no clearance. They don't respond to any of the stress conditions. as opposed to deamidation, where the source is the bioreactor and potentially in the downstream process based on temperature and pHs. Could be stability indicating if it's a liquid hold and there really isn't any clearance. And again, we start to build out what the process controls are in terms of parametric and material attribute controls, and what analytical controls should be in place, and how this impacts the residual risk. And as we mentioned before, for the high manos, the process capability is low for the dynamic perfusion example, high in steady state perfusion. Again, this was just invented data. We're really looking at a condition where you may have more variable or less variability in that attribute, and with no downstream clearance, you're really relying on bioreactor control. And as part of that control strategy, we also put together tables with some typical parameter ranges and how those may be altered as you move from the initial development and characterization through clinical manufacturing, through PPQ to commercial. Excuse me. And also included some events that may have occurred during clinical manufacturing, for example, a variation in trace metals that impacted glycosylation, which led to a tighter control over certain individual metals in the incoming cell culture media. Some variation that was seen in a downstream resin and variable performance over the initial pH range tested with one lot of resin, and how that impacted a tighter control in that step in the latter stages of the control strategy. So in commercial control, you had a tighter pH range. And finally, there was an event we talked about in the case study where you had a longer pause in the process during one of the clinical runs, going back to the lab to support that with additional small-scale data. That range was extended for the commercial controls. So there are some, I think, realistic examples in there of how control strategy is shaped not only by the initial process development, process characterization, but events that occur as you are experiencing more variability in raw material lots, possibly some variability in operations, and what can be supported, and how that control strategy will be adapted to provide the adequate control of the process. And we also include a chapter on adventitious agent control strategy, and really highlighting three elements of that: prevention, removal, and detection. So the two elements of control are really prevention and removal. So minimize the potential for introducing contaminants and verifying the capacity of the process to remove or inactivate potential contaminants. And the detection piece is really verifying that your control strategy has been successful. This could also be considered a way of verifying that you are operating in a closed operation. And going along with that adventitious agent control strategy, we put together a prototype bioburden sampling and testing plan. Again, the viral testing is pretty well established in terms of harvest testing, leading to the enabling the release of material. But for bioburden testing, we put together a fairly comprehensive plan, but put in elements of risk so that could be reduced in the frequency of testing for lower risk elements of the process. So for example, in the polishing purification steps where you may have more stringent cleaning protocols in place, may not need a very high frequency of sampling in an extended run. But again, we put this here to really drive some discussion. We found that having the discussion, writing things out was very useful. Putting together an image and an actual table laying out a sampling plan drove a lot more discussion, which is why we tried to include that. And we also included considerations for bioburden deviations in that intensified continuous bioprocess framework, so that you may have different limits in place in process, a reject limit, an action limit, alert limit, and how you would react. And in the case study, we did consider only traditional types of processing, where you would have a 2 to 3 day delay in getting your bioburden result. So it's a challenge when you have a process that's continuously moving, as opposed to batch where you could get a result before moving on to the next step. There will be some follow-up work as part of the process intensification NIIMBL-led program to look at the impact of rapid bioburden testing on those in-process decisions related to bioburden quantification. And again, we had a similar decision tree for the types of in-process decisions you may need to make in terms of assessing deviations. And here's a decision tree for CPP deviation, and trying to understand the root cause as well as the impact, and how those two assessments come together to drive a decision to either continue processing, segregate a portion of the batch, or divert to waste. Similar to the bioburden decision tree, we thought it was useful to actually include that decision tree as part of the case study. These often occur in organizations. They're not always very explicitly documented. And actually trying to get an explicit documentation of what that decision tree looked like did take a certain amount of effort. But we think, again, it's useful to bring this picture into play because it really highlights, I think, much better than several paragraphs of text, how you really make a decision, and as we add on layers later of how quickly one needs to make that decision. And then in the final chapter, we talk about some future directions, a more expanded PAT strategy, and two elements to that. It really requires different technology domains. So you certainly need the technology around the sensor, either the inline sensor or something that's available at line, but able to still give you rapid results. But that needs to be inserted into a robust digital environment so that you can manage the data, enable the data to be stored in a easily retrievable manner that's hopefully consistent across sites, and the ability to rapidly evaluate that data, comparing it to past history, whether coming from process characterization or process characterization enriched by all of the process data that has been generated in between those early studies and when deviation may occur, and whether or not you would need models to support that, AI or machine learning components, maybe a hybrid model that's partly empirical, partly mechanistic, partly machine learning. and the ability then to control the process within the limitations of the dynamics. When do you intervene? And we do talk about both a sampling point and a defined intervention point. And again, as I mentioned before, creating robust and reliable process models to predict those responses and synchronize any changes and represent them in a reasonable way so that you can assess it within the quality management system of a given organization. And we also go on to talk about additional information to establish that effective control strategy. And that's where we left things at Future Directions. Thank you for your attention. And Sandeep, I think we have a little time for a few more questions.
Yes, James, we have a few questions here on chat, and please, I remind folks to post your questions on chat so we can ask them, or we can get back to you if you don't have enough time to be asked here. So one question that came up, Gene, is around technology, and folks are curious about knowing, are there opportunities to evaluate next-gen technologies within this framework, such as other purification techniques, for instance?
Yes, and I'm hesitating a little. That can be done both as a paper exercise, as a case study, and we're looking at ways to kind of divide this what had become a rather large case study into smaller bites. So we are in the process of publishing at least 3 journal articles, one with an overview of N-mAb, one focusing on the process characterization and early validation work, and a third focusing on real-time decision-making, real-time process control. There's also a potential extension to what we have in N-mAb, as I mentioned, around real-time bioburden control and how that impacts your decision-making. And I think there could be opportunity, as was the case with A-mAb, for additional papers that come out of this. The second opportunity is an actual physical manifestation of this equipment train. And as many of you may know, NIIMBL is putting together a intensified continuous bioprocess testbed at the University of Delaware. And the idea is to set that up both to operate in a standard mode, but really provide the opportunity to test different unit operations, different sensors, how those would interact with downstream controls. As I mentioned, are the dynamics right to actually get effective control? So I think both are important. One is the paper study, expanding the conversation, and secondly, to generate actual data in a physical test bed. Next.
Thanks, Gene. And related to that, will NIIMBL conduct an evaluation of available in-process measurement resources, contrasting current probe reliability versus industry needs?
Yes, that is the plan. And I'm thinking that John Erickson may address that in his webinar on the process intensification NIIMBL-led program. But that was really what the test bed was designed for, both to test different equipment as well as provide a training environment for people who want to learn how to operate, as well as potentially reviewers or inspectors who want to see what this looks like in practice before they need to review or inspect processes.
Thank you. Another one is basically, Gene, we may or we may not know the answer to this, as the information may not be publicly known, but do you have a feel for how many processes are produced in a continuous culture versus a fully integrated continuous process way?
Yeah, that's a good question. I don't know. I know that there are a number of approved products that came from continuous upstream operations. Many of the early products from Bayer, from Genzyme, from Centocor were all coming from continuous processes. And these were driven by either the attribute of continuous culture to reduce the potential for chemical degradation within the bioreactor by limiting its residence time, or the fact that your productivity per capital investment is pretty high with continuous perfusion operation, or even with a low producing cell line, you can still generate hundreds of kilos, tons of material in continuous operation. I don't know currently how many processes have a fully integrated downstream that links to that. I know there are companies like Sanofi and AstraZeneca that are looking at this very closely right now.
Thank you, Gene. Another question that just came in is, could physical testbed be used to propose a data-driven control strategy? For example, with this actual data, this is what the filing could look like for a BLA, for instance, especially moving decisions and control parameters closer to the point of occurrence.
Yes. Yes, it has that potential.
Thank you.
Again, the advantage, many organizations are probably doing this at the same time. The advantage of having the public-private partnership is that we can do this and generate that data, publish it, and make it known more broadly. Okay.
Thank you. And one more question that just came in. Could you comment on whether a discussion around drug product formulation was also in context when ICB was discussed?
It was part of the original discussion. We decided to leave that as a potential future chapter, and the impact of upstream drug substance, that is, continuous or advanced operations on drug product formulation and fill operations, as well as interaction with any devices. So that's a potential future chapter. Okay.
Thank you, Gene.
And again, as I mentioned, how to access it. You can use this web link here. If you have your phone, you can use the QR code. That'll take you to the same page. The N-mAb case study, again, is under the drop-down menu for technology on the NIIMBL website. And we will ask you to fill in a short summary of who's downloading it to access the case study. We also have the poster from the ICB meeting in early October that's available, which gives you a kind of a quick overview. Sandeep, do you want to go through this?
Sorry, I was on mute, Gene. Thank you.
Yep.
So that's all the time we have, folks, today for the questions. But if we didn't answer your questions, we'll get back to you in a follow-up email. I'd like to close by, giving you some updates on the members, how we can follow up on the NIIMBL projects. So the way it works is basically you will receive, you choose the programs or project you would like to follow, and you click on the page and the follow button on the center right there. And the way it works is basically you would get an email update when the follow-up of a project has been updated on the web portal. The email will have a link to the project that you decided to choose to follow up. and then you can click and read the updates. If you followed up multiple projects or programs, then you'll be sent one email for all the updates. Next page. So in closing, lastly, we'd like to remind the attendees that we'll receive a survey link by email shortly after the webinar. You can complete the survey for immediate access to the recording of this webinar, or the webinar will be posted on the NIIMBL N-mAb webpage later this month. Our next webinar on biomanufacturing readiness level will take place next Wednesday on December 14th, and you can register on NIIMBL.org. A second series of webinar series will take place in February and March of 2023, and will feature our process intensification and vaccine manufacturing programs, as well as other additional sessions on N-mAb. Please stay tuned for registration information. Lastly, I would like to thank the attendees as well as Jean for coming out and presenting the N-mAb case study. And please feel free to reach out to Jean directly if you have any further questions. Enjoy the rest of your day, folks.
Thank you very much.